Hey everybody, in this video I am going to talk a little bit more about nonsense mediated decay. Specifically, we are going to look at an example of how the cell can use nonsense mediated decay to limit the number of times a messenger RNA can be translated, and that way it can have a really strict control of the number of proteins that can be made by a gene after transcription. So nonsense mediated decay, I lost my brush, sorry. Nonsense mediated decay, oh my god, what is going on? Okay, there we go. Nonsense mediated decay, NMD. And I have to apologize. In I think the last two videos, I may have, well, I know I did a couple times at least, but I called the premature termination codons, I called them pre-termination codons, and it wasn't until I was loading the videos up to YouTube and I was looking at the title of the video and I saw that it was labeled pre-termination codon and thought, oh, that doesn't make sense. And then I realized, oh no, premature termination codons. That is a PTC, premature termination codons. So I apologize for that. It was correctly described in the notes, though, so maybe you didn't even notice. So just a quick review on NMD in case you are hopping into this video and you didn't watch any of the previous ones. So up here at the top, excuse me, I have a messenger RNA diagrammed. So this messenger RNA has the cap right here, has the cap binding complex bound to the cap. We have a poly-A tail, which is about 250 nucleotides long on messenger RNAs in humans. We have a start codon, we have a stop codon, and we have these things here. So this EEJ, that's exon-exon junction. And these pink things, they are installed 20 to 24 nucleotides upstream of the EEJ. These are exon junction complexes. So when this messenger RNA is exported to the cytoplasm, it will be translated. So when it undergoes translation, one of the first things to happen is the cap binding complex, CBC, is exchanged for a different complex called EIF4E. So here we have still the poly-A tail. I didn't diagram the poly-A binding proteins there. We have the stop codon, start codon. And here's our ribosome, and the ribosome has started translating this messenger RNA. So the very first ribosome to translate the messenger RNA is called the Pathfinder. Pathfinder ribosome. And so what this ribosome is going to do, it's going to travel down that messenger RNA and it's going to remove all of these exon junction complexes. So I have that diagrammed down here. So we can see that the exon junction complexes have been dissociated. The ribosome has traveled the RNA all the way down to the stop codon. The stop codon enters the A site of the ribosome. We didn't talk about the E and the P and the A sites in this course. That's more of a cell biology topic. So there are no tRNAs, there are no transfer RNAs that recognize the three stop codons. The three stop codons are recognized by Erfs, eukaryotic release factors. Eukaryotic release factors. There are 3 of these. And what the Erfs do is they recognize the stop codons and they help initiate translation termination. But there are a number of other proteins that associate with the Erfs. So I have 2 Erfs diagrammed here. And these 4 proteins that we have here, we talked about these in a previous video, they are combined with the Erfs to make the SURF complex. SURF, that name SURF is derived from the SMG proteins and a protein called UPF1. So what this SURF complex does is it searches for exon junction complexes. And if it doesn't find any exon junction complexes on that messenger RNA, well, then the messenger RNA is okay. It's not going to be degraded. And it's free for other ribosomes to latch on to that 5' UTR, 5' untranslated region and start translating at the start codon. So, however, if there is a premature termination codon on the messenger RNA. So here I have the PTC diagrammed here, upstream of an exon junction complex. I have the normal stop codon downstream of the exon junction complex. If a PTC is encountered by the ribosome, well, the eukaryotic release factors will recognize the stop codon. It will terminate translation in the SURF complex. We'll check for exon junction complexes, and if it finds one, well, it will degrade that messenger RNA. And this is widely recognized as a quality control mechanism. And it's thought that PTCs, premature termination codons, usually result if there's a mutation in the gene or an error by POL2 during transcription. So, and the thought is that the cell wants to get rid of those messenger RNAs that have a mutation, that have a PTC, because a truncated protein could cause a lot of problems. And I believe in the last video I went over an example of how a truncated protein could cause some pretty big problems for the cell. So that's how nonsense mediated decay works. It's thought to be a quality control mechanism. And why does it work? Well, it depends on the position of the exon junction complexes relative to the stop codon, right? The normal stop codon and potentially premature termination codons. So I'm going to zoom out a bit there. So let's look at the typical gene structure of a gene in eukaryotes like humans. So here's just a typical generic gene structure. We have the +1 site where the gene is RNA, where POL2 begins transcription. We have the transcriptional cleavage site down here. We have 5 different exons diagrammed here. And we have 4 introns. Now, the start codon is usually found in the first exon, and the stop codon is usually found in the last exon for most genes, say in humans. So when this is transcribed and that messenger RNA is processed, we end up with a messenger RNA that looks something like this guy right here. So we have the poly A tail, cap binding complex bound to the 7-methylguanosine cap. We have the start codon, we have the stop codon, and we have all of the EJCs upstream of that stop codon. Again, these are the exon-exon junctions right here. Those are the places where the introns have been removed from and the exons have been fused together. The EJCs are 20 to 24 nucleotides upstream of each exon-exon junction. So for most genes, this is great, right? Nonsense mediated decay won't affect translation or stability of this messenger RNA because the Pathfinder ribosome will remove all of these exon junction complexes during the first round of translation. And then when it stops right here, there are no more exon junction complexes on that messenger RNA. So this messenger RNA won't be affected by nonsense mediated decay. And the only way it would be affected, say a messenger RNA from this gene, is if there was a mutation, let's say here, that changed a coding codon to a stop codon, or if POL2 made a mistake and with one transcript accidentally put a stop codon or produced an RNA, with a stop codon where there should have been a start codon. So, okay, so most genes are okay. So nonsense mediated decay seems like a pretty neat way to, control for mistakes. However, there are some genes in humans and other eukaryotes which have a non-standard gene structure, which is different than normal. So the ARC gene, ARC, is an example. So the ARC gene has 3 exons. We have the transcription start site here. We have the transcription cleavage site here. And we have the start codon in the first exon as normal, but we also have the normal stop codon in the first exon. So normally it's over here in the last exon, but not for this gene. So this is transcribed in the pre-messenger RNA, is processed into an RNA that looks like this. Cap binding complex here, poly A tail with 250 nucleotides, A nucleotides, 2 exon junction complexes, and 2 exon-exon junction sites. And you can see with this messenger RNA, we have a problem, right? When the ribosome jumps onto this and starts translating, it's going to stop here. And that ribosome is going to detect, when the SURF complex joins the ribosome at the stop codon, it's going to detect the EJCs and it's going to degrade that messenger RNA. So this ARC gene can only be translated one time. Assuming nonsense mediated decay works normally. It is an efficient process. So at most one time. So each messenger RNA, we normally think messenger RNAs can be translated many times over and over again to make many, many proteins. In this case, this messenger RNA can be translated only one time. At least that's the common model of ARC gene translation. And so why would the cell do that? Or why would the genome encode a gene that can only be translated one time for each messenger RNA? It doesn't seem very efficient, does it? Well, it turns out with this gene, this is a gene, and as of, I think, 10 years ago when this study came out, researchers didn't know what this gene does. And maybe they know now, I'm not really sure. But the ARC protein is needed in the nervous system. So I think there are mutants, maybe mouse mutants in this gene that have trouble with memory. So it's somehow involved with memory and probably some other aspects of the nervous system. So the ARC protein is important in the nervous system, and it's thought that, okay, so this is my diagram of a nerve cell. So here, I'm not sure how much of, how much, I guess, neuronal biology you guys know. I don't really know any, but I know enough how to copy this figure from something I found on the internet, or use something I found on the internet to diagram a nerve cell. So we have an axon here. We have these dendrites. Dendrites there. And essentially, nerve impulses travel through the axons, and they can connect with the downstream effectors can be dendrites of other nerve cells, or they can be like muscle cells and things like that. So here we have an axon from another nerve cell connecting to a dendrite of this nerve cell. So this is going to be a synapse, and I've diagrammed the synapse over here. So it's thought that the ARC messenger RNA, when it's exported out of the nucleus, there's a protein that binds to it that keeps it from being translated. So it can't be translated yet. And that protein, not only does it keep it from being translated, it helps transport it to the synapses of associated with the dendrites of that nerve cell. So let me zoom in on the synapse over here. So let's see. Here's the synapse. Here is the end of the axon. Here is the dendrite portion of the synapse. And so there's some signaling that's going to happen between these when a signal travels down the axon. So I think you can make out my diagram here. So here's the ARC messenger RNA. It has not been translated yet. I have this protein bound to, say, the ribosome binding site, so it cannot be translated yet. Now, it's thought that something happens when a signal, when this axon signals this dendrite through the synapse, something degrades this protein here, or maybe phosphorylates it so it dissociates from the messenger RNA. And then ribosomes translate this messenger RNA right here and make one protein. So that one ARC protein, well, one ARC protein for every ARC messenger RNA is made right there in the synapse. And then that ARC protein can do whatever it is supposed to do. And then after that one round of translation, right, well, the exon junction complexes are going to be recognized by the Pathfinder ribosome, and that messenger RNA is going to be degraded. So this is essentially nonsense mediated decay, right? But the authors termed it, gave it a unique name, so translation dependent decay. But we can essentially use these terms interchangeably. I guess when the cell is using translation to purposely degrade a messenger RNA, because if it didn't do it, it would lead to bad things. We can call it TDD. Maybe NMD, we can use that when we're describing, say, premature termination codons as a result of errors by RNA polymerase II or mutations in a gene. Either way, we're okay if we use NMD. NMD is good for both of these. So that's pretty neat. So essentially, I want to add one more fancy term in here. Spatiotemporal control. So nonsense mediated decay can be used by cells to control translation of messenger RNAs in a spatiotemporal fashion. So in space and time. And how does that relate to what we just went over? Space. So where is the messenger RNA going to be translated? Well, in this case, it's not going to be translated until that messenger RNA is brought to, well, it's only going to be translated in the dendrites, right? At the synapse junctions. At the junctions in the synapses. So that's the spatial control, the Spacio control, or the control in space. What about the time? Well, it's not going to be translated until a signal has been transmitted down that exon. So pretty neat, huh? Nonsense mediated decay can be used to control translations of messenger RNAs in space and time, which is, yeah, a neat way to think of it. Normally we think, okay, messenger RNA is exported out of the cytoplasm, it's translated right away, and it's translated many times. So we're seeing that doesn't need to be that way. Cells have ways to control where and when messenger RNAs are translated. So one more thing I'd like to cover. So how common is this translation-dependent decay? To simplify things, we can just call it NMD. How common do cells use an NMD to control how many proteins are made from a messenger RNA? So another way to ask this question is, how many human genes have introns in their 3' UTRs, in their 3' untranslated regions, right? So what is the definition of a 3' untranslated region? So here's a messenger RNA, here's its cap, here's its poly A tail, here's its start codon, here's the stop codon. So that's the 3' untranslated region, so the region after the stop codon. So if we can identify all the genes that have an intron in the 3' UTR, well, maybe we can have an idea of how many genes are regulated by this nonsense mediated decay. So the authors here's the paper. You can see the full reference in the notes. Georgi et al., 2007. Or Georgi. I like Georgi better. That's a cool last name. So what they did, they found, and this is 2007, so we probably know more now, but in 2007 they found 44,000 predicted pre-messenger RNAs in the human genome. So the human genome, they identified 44,000 genes that can produce 44,000 different pre-messenger RNAs, and some of those are due to alternate splicing of the same gene. Then they selected, among those, they selected those that had introns in the 3' untranslated region, those that were predicted to have an intron in the 3' untranslated region. And then among those, they selected for conservation of the 3' UTR in both the mouse genomes, rat genomes, and human genomes. So all three of these genomes. And the idea there is they don't really want to find every one of these cases. They just want to see if there's, how many there are. And they really want to be sure that these 3' UTRs are important and have been conserved. I'm sorry, these introns in these 3' UTRs have been conserved between multiple species. And if they have, that suggests that that intron is important for regulation of that gene. At least that's the argument the researchers made. So then among those, they're going to select for genes with stop codons that were 50 nucleotides, at least 50 nucleotides upstream of the intron. And we saw this in the previous video. If the stop codon is too close to that intron, well then the ribosome will end up dissociating that intron before it reaches the stop codon. And make sure you watch the previous video if you're confused about that. And in the last step, they ignored any gene that if there was a 3' UTR intron, if sometimes an alternate splice variant of that gene did not have that intron in the 3' UTR, they ignored those genes. They would ignore those. So after all of those things, they whittled down they went from 44,000 pre-messenger RNAs to now I don't know if this is now I'm realizing that is this 149 genes or 149 pre-messenger RNAs, because we can have multiple pre-messenger RNAs from a gene, right, due to alternate splicing. So to simplify things, let's just assume we're using genes here. So they found 149 genes in humans that are controlled by TTD or NMD, or they predicted. I'm sorry, I should fix that. So their analysis predicts that there are 149 genes in the human genome that are controlled by translation-dependent decay, nonsense-mediated decay. And why is that? Okay, because 149 genes have what? Well, let me do the structure here. Here's a generic gene. Here's the start codon. There's the +1 site. There's the transcription cleavage site. I'm going to put stop codon here. And this is at least 50 bases, 50 base pairs upstream of the beginning of this intron. So 149 genes in humans have a structure like this, and this intron here is in the 3' UTR, right? Because if the stop codon is right here, then everything after that is going to be in the 3' untranslated region. And this intron is in the 3' untranslated region. And for 149 genes in humans, this same 3' UTR intron is found in the orthologous gene in mice and rats. So, okay, pretty neat, I think. Now, I don't know if anyone's actually gone to study those genes specifically and show that indeed they are controlled by nonsense-mediated decay, and each messenger RNA from these genes can only be translated one time before it is degraded by nonsense-mediated decay. I'm not sure if anyone has followed up on that, but it seems reasonable to me that at least some of these, if not most of them, will be degraded by nonsense-mediated decay after they're translated one time. So, okay, so that's it for this video in nonsense-mediated decay. Next, we are going to take a look at the mediator complex and enhancers and enhancer RNAs, some really neat things that are, rather confusing and a really hot area of research right now. And, well, I'll talk more about it in the next video.